So far in slide A, we have in the recordings for slides chapter 9, part A, we have discussed the events at the neuromuscular junction. And remember that had to do with the action potential arriving at the axon terminal of the neuron and calcium ions coming into the axon terminal, stimulating the release of acetylcholine into the synaptic cleft, and the acetylcholine binding to receptors on the muscle fiber led to sodium coming into the muscle fiber, and that led to the generation of an action potential in the muscle fiber. And then we talked about the propagation of that action potential down the length of the muscle fiber, down the sarcolemma and into the T-tubules of the muscle fiber. So now we're going to talk about what happens when that action potential actually leads to a contraction in the muscle. And that is called, the action potential part is the excitation part, and the contraction is the actual shortening of the sarcomeres when the myosin attaches to the actin and pulls the actin in closer together. So that's called excitation, contraction, coupling, the events that transmit the action potential along the sarcolemma coupled to the sliding filament theory, which is the contraction part. So let's move. There we go. Okay, this is the first picture of excitation, contraction coupling, and where I like this because you can see the axon terminal at the neuromuscular junction here. You can see that part. We've already covered the events that happen there. The neuron stimulating the motor end plate, or the stimulating this particular region of the muscle fiber, and that leading to depolarization to threshold. Now the axon potential is generated along the sarcolemma of the muscle fiber, and then it is transmitted or propagated down into the T-tubule of the triad here on either side of the T-tubule. Remember, there is terminal cisternae of the sarcoplasmic reticulum, and they are filled with calcium. They are calcium reservoirs. So what happens with the calcium? What does the calcium do? The calcium, once stimulated by the action potential, the calcium is released and binds to the actin, and it causes the active sites on the actin fibers. You can see them as little black dots. The active sites are exposed. The myosin, which looks like the part that binds to the actin, looks like kind of like a golf stick with two clubs on the end. Well, those two clubs of the myosin are going to bind to the active sites on the actin. This is all triggered by calcium ions. And then the myosin, once it's attached, will pull the actin filaments toward each other. And that will be the contraction part. Okay, so the excitation part is the action potential leading to the release of calcium ions, and then the contraction part starts with the release of the calcium ions and ends with the actin filaments coming together, shortening and producing a contraction. Okay, so when there's low calcium ion concentration inside the muscle fiber, tropomyosin is a myofilament that blocks the active sites on actin. The myosin heads cannot attach to the actin, and the muscle fiber remains relaxed. At higher intracellular calcium ion concentrations, the calcium binds to troponin, and binding to troponin, I can blow this up so you can see it. Troponin is this little structure in here, this little protein that looks kind of like, it's yellow and it looks a little bit like an upside down snowman. When the calcium binds to the troponin causes this filament called tropomyosin to move out of the way. It had been covering these active sites, but now it's moved out of the way, and these active sites are exposed. Myosin can form cross bridges and bind with the active sites. And now the myosin, once it's bound with the active sites on actin, can pull the actin in, creating a shortening of the sarcomere. Now let's just look at all of the events at one time. Okay, so step one, we have crossbridge formation. So what happened? Calcium came in. Calcium from the terminal cisternae was released into the T-tubules, or was released into the muscle fiber. The calcium bound to the troponin. The troponin moved the tropomyosin out of the way of the active sites, exposing the active sites. And now the myosin heads attach to the active sites and pivot, and this causes a contraction. Now, that contraction will remain locked until ATP is broken down. So what is the role of the ATP? Is to actually remove the myosin heads from the actin. If this doesn't happen, another contraction is not possible. So the ATP comes in and breaks down and gives the energy to remove the myosin heads from the actin. And now that the myosin heads are removed, that muscle fiber is ready to contract again, but it cannot contract again without removing those myosin heads. Okay, so look at the last few slides. There are some questions about rigor mortis. There's at least one on the exam. It says 3 to 4 hours after death, muscles begin to stiffen. The peak rigidity, which is what we call rigor mortis, where the body is actually very stiff, occurs about 12 hours after death, post-mortem. The intracellular calcium levels increase because ATP is no longer being synthesized. And what did we talk about would happen if we had high levels of calcium inside muscle cells? Calcium results in crossbridge formation. Calcium causes the active sites to be exposed on actin, and myosin can now bind to actin and form a crossbridge, leading to a contraction. But we can't get the contraction remains until the myosin detaches from the actin, and that can't happen without ATP. And a dead body is not producing ATP. So the muscles stay contracted until muscle proteins actually begin to break down and decompose. Okay, and that will cause the release of myosin from actin. And that's it. So I'm going to stop this one here at 8 minutes.